Well, thank you very much, Jeremy. It's a great pleasure to be here. I've been an admirer of this conference series for many years and watched many of the videos. You know, instead of watching videos of cats falling out of trees, I've been watching these. So I really appreciate the opportunity to come and contribute to this conference and show you some of our data from what I think is an interesting story. So this story's been rumbling on for about 4 years, and you'll see from the different types of graphs that I show that various people have been involved. And I'm going to tell you today about how human T cells can recognise unusual and unnatural peptide epitopes. Now, this all started with a hypothesis, and our hypothesis really was based around my background as a human T cell immunologist working in cancer. When you're in that field, you're always looking for tumour antigens, and we heard a great talk this morning about looking for neoantigens. But many tumour antigens are defined based on the fact that they're overexpressed in cancers compared to normal cells. For our working hypothesis, we took the opposite view, and we looked at things which had a lower expression. And that hypothesis is really summed up by this great world leader. And you may laugh. This was actually sent to me by an American colleague, and with hindsight now we may see these as the golden years. So we wanted to look at proteins which had a low expression, and the reason they had a low expression is because in cancer cells you get this process where proteins can be abnormally degraded as part of the cancer process, and there was a very nice review about this in Cancer Cell in 2003. And this is out of date now, the hallmarks of cancer, but the corruption of many normal cellular processes are due in part to abnormal breakdown of important proteins that control these processes. Now you might be thinking, well, what's this got to do with immunology? Well, it turns out that the mechanism used for this protein breakdown is the ubiquitin proteasome system. And of course this system is the system that generates peptides which can be loaded onto MHC class 1 molecules. So we reasoned that perhaps some of these peptides from these abnormally broken down proteins are making their way to the cell surface on MHC molecules and could be present on cancer cells, but not on normal cells, because this process is not happening in normal cells. So these proteins are ubiquitously expressed, but they're just abnormally broken down in cancer cells. So we focused first of all on a protein which is involved in apoptosis called BAX, which is a pro-apoptotic protein. And the reason we were interested in this is because there were reports in the literature that this protein was broken down in many types of cancer. And actually at the time, the one we were most interested in was prostate cancer. So increasing grades of prostate cancer, more breakdown of this protein. So we thought, oh, that could be a good tumour antigen and a target for prostate cancer. It turned out that it wasn't when we did it, but we did have a proof of concept study where we showed that T cells from a healthy donor generated against BAX peptides could recognise primary cancer cells from patients with leukemia, chronic lymphocytic leukemia. So we thought, great, that could be a tumour antigen. But of course the real proof of the pudding is to find those T cells in patients and to expand them. So this is where a PhD student, Rhys Reed, came in, and his project was to grow T cell clones from patients with leukemia against these BAX peptides. So this is a scheme, I think familiar to many people in the room, of cloning T cells. So here, instead of using some of the fancy proimmune assays, we did it a bit more low-tech and low-cost. We looked at candidate peptides using peptide prediction programs, and then we bought 23 peptides and used those as a pool to stimulate T cells. And then any T cells which were producing interferon gamma and ELISpot assays, we could isolate with magnetic beads and then clone them. And this shows you that we can stimulate peptide-specific T cells from patients. So Rhys was able to stimulate these T cells, quite low frequency, but able to stimulate them detectably in ELISpot assays. So these are interferon gamma-producing T cells. And you can see that there's some peptide-specific T cells detected, and also some T cells which seem to recognise the tumour cell without any peptides. So we were hoping that there was some overlap within these two populations, that we could generate T cells which were peptide and tumour cell specific. So Rhys then went on to clone those T cells, and this just shows that he selected a clone called 6C5 from the many clones that he screened by ELISpot assay. And he chose this clone because it was the fastest growing clone and the easiest to work with. So he mapped the peptide specificity. Remember, we stimulated originally with 23 peptides, and he tested that T-cell clone against the whole peptide pool, and then he split the pool up into pools of 4 or 5 peptides, and then he was able to map to one of those pools, and then 2 peptides came up in the subsequent screen. And it turned out that those 2 peptides had overlapping sequences. And actually, interestingly, this particular peptide sequence was not one of the best scoring peptides in the, predicted to bind to HLA-A2 in this case. So he did a dose response to find out which of these 2 peptides was the dominant epitope recognized. And from his dose response using an interferon gamma ELISA, it looked like it was the 9 peptide. So that was all well and good, but you might notice here that these peptides were not completely pure. For reasons of cost, and this is going to be a reoccurring theme in my talk, reasons of cost, we ordered peptides which were so-called immunograde, so not 100% pure, but around 70% pure. So to see which of these 2 peptides was really being recognized, we had, actually it was ProImmune, synthesize a peptide to greater than 95% purity, and we tested the clone with that peptide. Unfortunately, that peptide was not recognized, and you can see here that this peptide, which was 77% pure, was recognized. So there's a few explanations that we could use for this data. One was that perhaps this sequence was wrong in some way, but it wasn't, because ProImmune showed us the mass spec data and the peptide was fine. Well, the other issue could be that perhaps there is an impurity being recognized. Something in the 23% is being recognized by the T-cell clone. Now at this point, Reese nearly abandoned the project, because obviously he's got a clone now, and he knew it was a clone because he did some further work with monoclonal antibodies to show that there was a single T-cell receptor, the beta region. But he also showed at the same time that these T-cell clones were not recognizing the cancer cells. So they're recognizing an unknown peptide, probably in an impurity. So at this point he wanted to abandon the project and go back and make some more T-cell clones. But we persuaded him to continue, and fortunately he did, because we then enlisted the services of a peptide chemist in the School of Chemistry in Cardiff, James Redman. And it was great talking to him, because he could then help us find out what was in that impure fraction, 23%. And he used liquid chromatography mass spectrometry, which he's an expert in, and he showed that in both the peptide preparations, the pure one and the so-called crude one, that the main component was the right peptide and had the correct sequence. But when he looked at the impurities, he found actually that there were some impurities, actually even in the 95% pure peptide, that there were single amino acid deletions detectable in both samples. But there were more impurities, obviously, in the crude sample, and there were some which were unique to that crude sample. There were some hydrophobic peptides, and he knew they were more hydrophobic because of the way they luted on his C18 column, but also some impurities which had peculiar masses, or particular masses. And he proposed several potential impurities. One was potentially a peptide which was too long. So this was a 10-mer peptide with an extra lysine on the front. So this might have come about through insufficient purification during the peptide synthesis process. Another impurity that could come about through a butyl group being attached to the tyrosine residue in the middle of the peptide. And this could come about again during the peptide synthesis reaction. And last but not least, because these peptides were made by an FMOC peptide synthesis method, you could have FMOC stuck onto the end of the peptide as an impurity. So he tried to recreate the crude peptide synthesis method. So here's the difference between the two results. This is the crude peptide with impurities, and here's the pure peptide. And so he made various preparations where he configured them so that he either only had the crude 9-mer, and then he made some deletions coming from the hydroxy end, and then some deletions going the other way. And then he also made the 10-mer peptide. But we didn't see any difference between these, and so we concluded that there must be something common to all of those crude preparations that's responsible for the T-cell recognition. And so then James took the crude preparation 1 and fractionated into 8 different fractions, and then Rees tested it in an interferon gamma assay with his T-cell clone. So again, you see that the crude preparation is recognised well, the pure not recognised, and then really fraction 8 is the fraction that's being recognised. And what he found that was common to both of those fractions, the crude peptide and fraction 8, was the butylated peptide. So this had a butyl group stuck on at position 4. So that's circumstantial evidence that a butylated peptide, which is present as an impurity from peptide synthesis, was the peptide epitope that was being recognised. So to prove it, James then took the purified peptide. So remember, this is not recognised. And then he deliberately added a butyl group to the tyrosine using 2 types of chemical reaction. I'm not a chemist, so I'm not going to go into them, but I think one of them involved blasting the peptide with butane gas. So he adds a butyl group to the tyrosine, and then Rees tested those for recognition. And lo and behold, the ones which had been treated with the addition of the butyl group were recognised. And James confirmed that these were in fact butylated through mass spec analysis. So there could obviously be something odd about the way we do experiments in Cardiff, or something in the water. So we decided to actually go one step further and have an independent company make a butylated peptide for us. So according to James, you can get tyrosine residues, you can buy those with the butyl group already stuck to them. So we used those, and we asked a company in France to make a synthetic peptide which incorporated a butylated tyrosine residue. And what they showed, they did make a peptide to 95% purity, and what these graphs show is that peptide does indeed stimulate the T-cell clone with great potency. So you can see with interferon gamma assays, you get a good recognition with the butylated peptide compared to nothing with the pure. And the same thing with looking at assays where we're secreting cytotoxic granules, the CD107 assay. And James estimated that in this crude population, peptide preparation, the butylated peptide was present at about 1%. And so when Rees did these dose response curves, it looked like that was consistent with that the butylated peptide was present at a very low level in the original crude preparation. So we have a peptide, and we estimated that when we stimulated with the 23 peptides, that contaminant was present at probably less than 0.05%, and yet it was immunogenic. We knew there were peptides in that pool which were predicted to have very good binding for HLA-A2. So why was that peptide recognised? So to provide some clues to that, we enlisted the services of Pierre Riscala, who is a structural biologist, and he modelled this butylated peptide and HLA-A2 complex. And what he was able to show was that the butylated peptide residue at position 4 sticks out of the HLA class 1 binding groove. So it's likely to be a T-cell receptor contact point. And the other thing that his model predicted was that there's at least 2 possible configurations of the peptide based on the rotation of the tyrosine with the butyl group attached to it, as shown in this figure. So there's 2 possible configurations. His model also predicted that you might be able to substitute for this butylated tyrosine by adding a bulky amino acid, such as tryptophan. So just to summarise this first part, we made some T-cell clones from a patient, stimulating with a mixture of peptides, and it turned out that the peptide that was being recognised was an unnatural peptide. Butylated peptides don't occur in nature. They only occur as part of the peptide synthesis process. So just by adding this butyl group to this peptide, you can get T-cell recognition. We don't think it was to do with better HLA binding, so adding the butyl group doesn't increase HLA binding because both these peptides, it turns out, are very poor binders. And the computer model does suggest that the butylated peptide is an important T-cell contact residue. And the actual contaminant was present at very low percentages, which obviously has some implications for using peptides which are not completely pure for your experiments and perhaps immunotherapy. So a lot of questions came up out of this, and one I've asked already is, why is this particular peptide immunogenic when it's got this butyl group stuck to it, which is unnatural? We can see that if you take the butyl group off, you lose T-cell recognition. So does the T-cell receptor actually have specificity for the type of chemical modification? Could we add other things besides butyl and get recognition or not? And then another question really was, this is a T-cell clone which is recognising an unnatural epitope. What the dickens is it actually recognising? So we then went to the question about, what is this T-cell clone recognising? It's a T-cell clone from a cancer patient, doesn't recognise cancer cells, recognises this unnatural peptide epitope. How do we find what it's recognising? So there we collaborated with Professor Linda Waldridge in Bristol and her postdoc Anya Lucina. And while Linda was with Andy Sewell in Cardiff, she developed this combinatorial peptide library screening system, which is a bit of a mouthful, so I'll just call it CPL from now on. And what this involves is screening T cells, and it's best to screen T cell clones with a library of 480 billion peptides. And this library consists of sub-libraries, 180 sub-libraries. Each one of these sub-libraries contains a series of peptides which may have one position fixed, and then all the other positions have all the different types of amino acid that could occur at that position. So you test your T-cell clones against these libraries, which have got all these different peptide combinations. So this involves using an ELISA, a MIP1 beta ELISA. So you mix in with your T cells, antigen-presenting cell, and your peptide libraries, and then you screen for MIP1 beta secretion. And then what you get out is basically a list of preferred residues at each, preferred amino acids at each residue, positions 1 to 9 of the peptide. And I'm going to come on back to this in a bit more detail in a minute. You can use the results from that to do a database search, and in this case we did viral and self peptides. And then that generated a lot of peptides, 400 peptides. So then we looked further at peptides which were predicted to be good at binding. And again, for reasons of cost, we restricted to 40 peptides in the end to screen. So this just shows you that middle part in more detail. So position 1, you can see there's quite a bit of degeneracy. This T-cell receptor doesn't mind, it's not too fussed about the different amino acids it's recognizing. In red is the amino acid of the actual peptide, the index peptide. So position 1, not too fussed, whereas you look at position 3, there's only 2 amino acids being recognized. In position 4, interestingly, only tyrosine and tryptophan are really recognized. So you get a list of preferred amino acids at each position, and then you can generate from that a T-cell receptor footprint, which is quite useful for comparing the reactivity of different T cells against the same peptide. So this sort of heat map shows you the regions where there's most variation or least variation. And you can also generate an optimal sequence. And if you're lucky, that optimal sequence might match something in the database. In our case, we were unlucky and it didn't. So Linda did use this method to define peptide epitopes, viral epitopes for T cells from a leukemia patient. And others have used it to look at the extent of cross-reactivity for T cell clones. So, as I said, we can use the results from these library screens to then search databases for different peptide protein sequences. So this is what we've got in a nutshell. I mentioned that we had 40 peptides, 11 viral sequences from common viruses such as herpes simplex and vaccinia viruses, or they were common when they were used in smallpox vaccination, but not so common now, and cytomegalovirus. And you can see in this MIP1 beta ELISA screen that we don't get much reactivity in the viral peptides, which is what we were hoping we would get a positive response there. We modified some peptides as well, so don't worry too much about that. But look, here's all the self sequences, or sequences derived from self proteins, and we're getting quite a few hits. Just as a little detour to go back to peptide purity again, the company that made these peptides, and it wasn't ProImmune, could only synthesize 2 of the peptides to I think it was 30% and 70% purity. So we tested those peptides anyway, while the company carried on and made purer versions. And you can see here that the 2 peptides, so this would have been a positive in our screen, and then once we get a pure version of it, we lose the reactivity. And same here, this would have been positive in our screen, and then we purify it and lose reactivity. And James actually went back and looked at those samples by mass spectrometry and found actually that they did contain peptides with a butylated tyrosine. So, but you notice that these sequences are different from the sequence we saw earlier. So it suggests that actually you can confer reactivity by adding that butylated tyrosine. So again, this is something we haven't followed up. It does illustrate the importance of peptide purity. So going back to that big list of peptides we had before, we selected 14 peptides and did a proper dose response on them. And you can see that there's a hierarchy of potency for MIP1 beta secretion. Here's the T-bu peptide, which is how we define the clone specificity initially. And you can see there's at least half a dozen peptides derived from self-protein sequences, which are much better. And some of these actually are 500 to 600 times better when we compare the doses. So, where does that leave us? We have a T cell clone, and it recognizes this unnatural peptide, and it also seems to react on all these peptides which are derived from self proteins, and some of them are very potent at inducing T cell function. I can't tell you that the T cell clone is autoreactive, because we don't actually know that these peptides are actually processed and presented by T cells, but it does look like that some of these peptides have got features in common. So there's an alanine or glycine at position 3, tryptophan at position 4, and then a phenylalanine in position 6. So these are the sorts of things which were predicted from that peptide screen I told you about. The other thing you notice is that if you compare that sequence there, these sequences are all really dissimilar. So it's not like they're related in sequence. They may be related in structure, and that's something we plan to investigate, but they don't seem to be related much in sequence. Only 2 amino acids residues in common. So, conclusions. We've identified this unnatural T cell epitope, which seems to be a consequence of peptide production. What we've shown is that the T cells do seem to care about what type of chemical modification they encounter. So the T cell receptor seems to be able to distinguish between different chemically modified residues. And then we use this combinatorial peptide library. Now, we didn't manage to define a bona fide epitope. Most of the epitopes seem to come from self-derived proteins. And they are unusual in that respect because we were hoping to define some viral peptides. But interesting thing was that all these peptide syntheses are quite dissimilar compared to this T-bu peptide up here. So what we think is going on is that these activation CD8 T cells with chemically modified peptides has got the potential to generate unexpected and unwanted T cell specificities. And I think this has a few implications. First of all, we have to worry about peptide purity if we're using peptides for experiments in the lab or perhaps for therapeutic purposes. And then the other thing that occurred to us is that if we can activate T cells by adding a chemical modification to a particular residue within a peptide, then maybe we can also turn off functions by the same criteria, and that's something else we're interested in too. So potentially we could use this thing to our advantage. Perhaps if you had a T cell recognizing an autoimmune peptide epitope, maybe if you chemically modified those peptides, you could turn off those T cells. And I'd like to finish now by acknowledging all the people who helped with this work. This is a typical sunny day in Cardiff. This is the Institute of Cancer and Genetics. And Reese Reed was the PhD student who's now at University of Pennsylvania. Shimaz was a postdoc working in the lab. And Debbie and Beth in different parts of the T-cell assay. Indebted to James Redman, who did with peptide chemistry, and Pierre Riscala, who helped with the structural biology, and to Linda and Anya in Bristol. And Ilona was a pharmacy student from the University of Utrecht through the Erasmus scheme. So these are all the funders of the work. So thank you very much.
Thank you.